Students, if you remember in our last class under chapter sexual reproduction in flowering plants, we were discussing about double fertilization. We will continue that. Double fertilization I already told you, and you have already learned in first year PUC also in detail. Double fertilization you have learned. So double fertilization and triple fusion is a characteristics of angiosperms, because this type of fertilization is not there in any other organisms. Even in other plants also this type of reproduction is not there. Only in angiosperms, that is angiosperm means flowering plants, only in the plants which produces flowers, only in those plants fertilization is called double fertilization, triple fusion. If you remember, already we have learnt in last class. Once again we will see what we have learnt. Okay, so this is, suppose if you remember, this is a ovule, and this is the embryo sac. These are the integuments. If you remember, this is the nucellus. So this is the embryo sac, you know. Embryo sac. Yes, sac means bag-like structure which contains embryo. So embryo sac. Then here, if you remember, there are 3 antipodal cells. Then here, egg will be there and 2 cells surrounding the egg, that is called synergids. So I can show egg in orange color here just to make you identify which is egg. Okay, this one is egg, which is present in the center, and these 2 are synergids. You know that. Synergids, if you remember. Then in the center there are 2 nuclei present. They are called polar nuclei. You know this. This may be more than 10th time you are learning this same concept. Okay, then polar tube. When the polar tube tries to enter, what happens? You remember? I will just magnify this much part and I will show you. Okay, so enlarged part. That is, see, this is a micropyle integuments, and here there is an embryo sac. Embryo sac is there, and this is egg, and on 2 sides there are synergids. If you remember, we have already learned in class. This one is egg, and these 2 are synergids. You already know this. Then, in the synergid, there is finger-like structure here. What is that called? Finger-like structure. That is called filiform apparatus. This is already we have learned in last class. Recap. Filiform apparatus. These 2 are synergids. This is egg and this is filiform apparatus. Then here there is polar nuclei. 2, same thing, same diagram, enlarged part, this much part enlarged and showing. Okay. So the same 2 synergids here, 2 polar nuclei here, this egg what you see, and it's here, and these 2 synergids you can see here. Okay. How exactly double fertilization occurs, we will see. So when pollen tube has to enter, suppose pollen tube is a pollen tube, it will have 3 nuclei totally, because you remember generating nuclei divides into 2. These 2 are called male gametes. Remember male gametes. Then behind that, 1 vegetative nuclei will be there. Vegetative nucleus. These 2 are called male gametes. Okay, so now when this pollen tube enters through micropyle, this is opening called micropyle. If you remember, pollen tube will enter this micropyle and tries to meet the egg, but directly it cannot meet the egg, because you know yesterday you have learned, and the pollen tube goes like this. First, directly it cannot meet the egg because it should enter synergids. In the synergids, there is finger-like structure. I am showing in yellow color. That finger-like structure it should enter. That is what filiform apparatus. Okay, so filiform apparatus function is to guide the pollen tube towards egg. Guiding means filiform apparatus will help the pollen tube to enter. It will guide the pollen tube how to go. Like if a new person comes to one area, he will ask where is this address. So then that person should show where exactly it comes, where exactly it is located. So like that, when pollen tube tries to enter, at that time filiform apparatus will guide the entry of pollen tube, where exactly pollen tube should go. So first pollen tube will enter filiform apparatus, then from the filiform apparatus, through filiform apparatus it comes to cytoplasm of synergid. Cytoplasm of synergid. Suppose this is one synergid cell. Here filiform apparatus is there. Okay, the pollen tube will come like this. It will first enter filiform apparatus, then through filiform it goes to cytoplasm. This is cytoplasm of synergid. Then what happens? The two male gametes, what is there here? Here are two male gametes and one vegetative nuclear is here. That is this. These two, what you see, this here, two male gametes. So two male gametes will be released into the cytoplasm of synergid. It is released into cytoplasm of synergid cell. So that these two nuclei, what is there, they will be released into cytoplasm of synergid. Then from there, these two nuclei, see, now it is released here. Okay, then what should happen? First nuclei, what is there here? First nuclei, the first nuclei, what is here? Now that will meet the egg, will meet the nucleus of egg. First nuclei will meet the nucleus of egg. Then what happens? Then this part becomes a zygote. Zygote. How zygote is produced? This nucleus, also this nucleus meets the egg. Okay, this one meets the egg. Then what happens to this egg? This nucleus meets the nucleus of egg, so the two nuclei fuse and it becomes this entire part becomes a zygote. See, entire part becomes a zygote. What is the ploidy of zygote? Ploidy means how many sets of chromosomes. Already this nucleus is N haploid, egg is also haploid. Then when this N meets N, it becomes N plus N, 2N. So zygote will be diploid, 2N. This zygote will be diploid later. Okay, this egg becomes zygote. Then what about the remaining part? What about this remaining part? Only this much part we have learned. What about the remaining part? This remaining part where one nucleus met the egg, what about other nucleus? Other nucleus present here, it will go and meet polar nucleus. One met here, other one will go and meet here. So that what happens? Already polar nuclei are there, each one is N. This is also haploid, this is also haploid. Okay, then along with that one more haploid will come because second one was there here, that will go and meet here. This is also N, then what should happen? N plus N plus N, because each polar nuclei is haploid, then one more, this nuclei also goes there here. Then what should happen? N, this is N and this is N plus N plus N, then this becomes structure which is 3N, diploid. What is that diploid? It is called N. That is primary endosperm nucleus. This will become primary endosperm nucleus. So that I can clearly show you instead of drawing so many diagrams, one simple diagram I will draw. What happens during fertilization? What is double fertilization? In a simple diagram, we can show. Suppose this is just an embryo sac, Okay, here one egg is there. Okay, or else you can show here also. Here one egg is there and two cyanogens are. Okay, so the center one is egg and these two are cyanogens. What happens? Let us see. The first nuclei which was there, first male gamete will meet the egg, and this entire part, orange color now, this entire part becomes zygote, which is 2N. How it is 2N? Because the zygote is produced by fusion of one male gamete and egg. Egg is also N, male gamete is also N. N plus N, both will combine and become 2N. So this entire part becomes zygote. What about the remaining part? Here, two polar nuclei were there. Okay, each polar nuclei is N haploid. Then later, during fertilization, these two nuclei will combine. They will combine and become 2N. That is called secondary nucleus. So instead of going in detail about secondary nucleus, simple terms we can say already here two nuclei are there, polar nuclei. Each one is N and N. This also haploid, this also haploid. Then one more new came here was two male gametes will be there in the pollen tube. One with the egg, other one will be here. So this is also N. So this whole part, what is there? This whole part, okay, this will become one nuclei now, N plus N plus N. So this whole will become one nuclei, that is called, that one nuclei, what is that? That will be called T-N structure diploid, and this nuclei is called primary endosperm nucleus. Primary endosperm nucleus in short pen. D, E, and pen. Okay, this is a nucleus. Usually, nucleus will be there in one cell. No? So, if this is a nucleus, then what is this cell? This is called PEC, primary endosperm cell. Which one? Except this zygote. Remove this zygote, right? Zygote already is formed. Except this orange color, remaining all the parts, it is primary endosperm cell. Okay. So this is when formation of pen, PEC, zygote, that is after fertilization. So the stages what we have to learn for formation of primary endosperm nucleus, primary endosperm cell and zygote happens after fertilization. So it is post-fertilization stages. Now what we are learning is fertilization. What happens? The first nucleus of the first male gamete of the pollen tube will be the egg, that is first fertilization. And second nucleus of the pollen tube means the polar nucleus, that is second fertilization. During that process what happens? Slowly, your three antipodal cells are there. Slowly antipodal cells will become smaller and smaller and they will disintegrate, disappear. They become smaller and smaller. Okay, so that is disintegrating antipodals. Meaning after fertilization, antipodal cells are not required there, no function. So antipodal cell will become smaller and smaller and it will disintegrate. Then what happens to these cyanogens? Because already cyanogid function is over. What is the function of cyanogid? To guide the entry of pollen tube. Already pollen tube entered, fertilization happened. Then what is the use of cyanogid? No use. Cyanogids present on either side of zygote now, that also will disintegrate. Cyanogids also disintegrate. Antipodals also become smaller and smaller and disappears. Cyanogids also become smaller and disappears. Only what remains is zygote, nucleus called primary endosperm nucleus, which is 3N, triploid, then one entire big cell, that is primary endosperm cell. The formation of primary endosperm cell, primary endosperm nucleus and zygote occurs after fertilization. So we are going to learn now post-fertilization changes. Till now whatever we have learned is fertilization, double fertilization. Why it is called double fertilization? Because fertilization happens 2 times. That's why it is double fertilization. First fertilization is when colon tube contains 2 gametes. First one means the egg and produces zygote. Second one means the polar nuclei and forms a primary endosperm nucleus. Then the fertilization is over. Next what we are learning is post-fertilization changes. But in textbook you see that along with this some diagrams are given. That diagram is related to next heading, that is embryo. Okay, so in explanation they are showing figure 2.13. Whatever they are showing diagram here, that is related to embryo formation. So next what we are learning is now what happens after fertilization, that is called post-fertilization. After fertilization, what happens? What is produced? That is post-fertilization. Post means after. After fertilization, what happens? Post-fertilization events or changes. What happens? After fertilization, what is produced? There will be formation of endosperm and there will be formation of zygote. No? Where ovules will become seed after fertilization. Ovules will become seeds and entire ovary becomes fruit. If you remember, this is a Ganesh ovary style sigma. Remember ovary style sigma? Yes. This ovary style sigma. This is a pistil or Ganesh. No, you remember pistil or also called Ganeshium. After fertilization, what happens? Inside the ovary, there will be ovules. These are the ovules. What is here? Ovules. So these ovules later becomes seed after fertilization. After fertilization, I was saying after fertilization, after entry of colon tube into these ovules will become seeds. Then entire ovary becomes a fruit. So this is after fertilization. So after fertilization, seed and fruit will be produced. And before production of seeds and fruits, there will be formation of endosperm and zygote. Just now, I was showing you in the picture. So these are all formation of endosperm, formation of zygote, and formation of ovules into seeds, ovary into fruits. All this happens after fertilization. So it is called post-fertilization changes. Post means after fertilization. Okay, let us see what happens. Now it is very interesting to think. That is endosperm formation. Why I say very interesting to think? Because now whether ovule development happens first or endosperm development happens first. There is one beautiful sentence given in the textbook. Endosperm formation precedes seed formation. Why? Because before production of seeds, endosperm should be developed first. Why? Because for development of zygote into seed, for that food is required. Food is provided by endosperm. That is why endosperm should be formed first. Then zygote will become seed. Yes, that is a case. Okay, so let us see what happens exactly. So first, there will be, this is a embryo sac. If you remember, this is embryo sac, and here there will be egg, and these are two synergies. No, synergies are disintegrating. Disintegrate means they become smaller and they disappear. And this is a egg. Now, after fertilization, what happened? This became zygote. This diploid 2N. Why it is 2N? Because zygote is produced by fusion of male gamete, first nucleus with egg. N plus N became 2N. Then here polar nuclei will be there. Polar nuclei later, each polar nuclei is N and N, and then one more nuclei comes from polar tube, that is also N, so N plus N plus N, and finally it produces one structure that is PN, diploid, is called PN, pen, that is primary endosperm nucleus. Primary endosperm nucleus. Usually nucleus will be there in the cell, so this big cell, what we call PEC, primary endosperm cell. So this primary endosperm cell will contain a nucleus in the center. That is primary endosperm nucleus. Primary endosperm cell. That means this big cell, what is there? The whole cell, it is called primary endosperm cell, and the whole cell contains one nucleus in the center, that is 3N, that is called primary endosperm nucleus. So the cell contains a nucleus. Then here antipodal cells are there, they are disintegrating. Disintegrating antipodals. This is when after fertilization. Once fertilization is over, egg becomes a zygote. This is a primary endosperm nucleus, and the entire cell is called primary endosperm cell. Okay, do you remember? Yes. Now my question is, why zygote should divide? Zygote should divide by mitosis. But before zygote divides by cell division, first endosperm nucleus divides. Both should divide. Zygote also should divide, and the primary endosperm nucleus also should divide. But now, interesting fact to note is this pen, primary endosperm nucleus will divide first, zygote will divide later. Why? That is a question. The statement given in textbook is endosperm development precedes zygote development. Before zygote divides and develops, endosperm develops. Why? Because for zygote to develop, it needs a food. For division of zygote and for development of zygote, it needs a food. For food development, endosperm should first form, because endosperm is a food for zygote. Okay, so that is the reason why endosperm nucleus divides first, pen divides first. After formation and division of this pen, endosperm is produced. Then, once endosperm is produced, then zygote will start dividing. That is the reason why this will divide first, because zygote requires food for their next development. How they will get food? They will get food only when food is produced there. So that is why endosperm divides and develops first, then followed by zygote. Okay. So what exactly happens? We will see now. Okay. There is a chalk. Okay. Sorry. This is a problem when we are recording with mic. Recording with mobile. Okay, what exactly happens? We will see now. These many changes are not required here now. I have to show only pen. Because we have to show only now how exactly this pen will divide. So this primary endosperm nucleus, what is that? Pen. This is diploid. First it will start dividing. how divides continuously means this primary endosperm nucleus divides first. Then after finishing division, cell division, then zygote will start developing. Because see now I'll make it very simple. Primary endosperm nucleus also should divide. Zygote also should divide. Which divides first? Primary endosperm nucleus divides first. Why? Because later after division it produces an endosperm. And endosperm is a food for zygote. Zygote will divide only when food is available. So that is why endosperm should divide first and formation of endosperm should happen first. So primary endosperm nucleus, this one nucleus divides into many nuclei. So many nuclei will be produced. So this is a primary endosperm cell. We will see that is this big cell. This nucleus will divide and produce many nuclei. It produces thousands of nuclei. We believe diagram is not there in the textbook. This is for your understanding. Thousands of nuclei, and all these nuclei are separated from each other. They don't touch. That is why it is called free nuclear endosperm. Free nuclear. Name only says all the nuclei are free. How many nuclei will be produced? One pen, that is one primary endosperm nucleus, divides continuously and produces thousands of nuclei. So one cell contains, primary endosperm cell contains thousands of nuclei, and all the nuclei are separated from each other, so they are separate. That's why it is called free nuclear endosperm. Example of free nuclear endosperm is they will be in a liquid state. That is tender coconut water. ನೀವು ದಕ್ಷಿಣ ಕನ್ನಡದಲ್ಲಿ ಬೊಂಡ ಅಂತಾರಲ್ಲ, ಬೇರೆ ಕಡೆ ಎಳನೀರು ಅಂತಾರೆ. Okay, so tender coconut water, what is there? When you drink tender coconut water, you are drinking free nuclear endosperm. Because when these free nuclei are there, thousands of free nuclei, it will be in a liquid state. So that is example, tender coconut water. Tender coconut water is free nuclear endosperm. Later what happens? Slowly, because you have seen that first when it is very tender, ಅಂದರೆ ತುಂಬ ಎಳೆದಿದ್ದಾಗ ಅದರಲ್ಲಿ ಒಳಗಡೆ ತಿನ್ನಲಿಕ್ಕೆ ಏನು ಇರೋದಿಲ್ಲ. Nothing is there to eat there when it is very tender. Only liquid will be there. So that is what we call free nuclear endosperm. And what tender coconut water, what you are drinking, that is free nuclei of produced by pen. Later what happens? See, the tender coconut doesn't remain tender only. Then it will grow. Then what happens? You ask something called something to eat inside. When tender coconut is broken, and you drink that water and you break it, then you see that a thin white color that is called caramel. Slowly what happens? Slowly each nuclei will be surrounded by cell wall. Slowly each nuclei will be surrounded by cell wall and they become a cell. So slowly what happens? Some of the free nuclei present on the sides will start producing cells here like this. That's why tender coconut, when you see first tender coconut, when you break open, remember this is the shell, the outer shell. So this is suppose inner area. First when free nuclear endosperm is there, it is only water, tender coconut water. That is what we call free nuclear endosperm. Slowly some of the nuclei will develop cell wall and they become cells on the sides. So what happens later? On these sides, here, there will be formation of cells. So this is called cellular endosperm. What is this white color? Which is they scrape it and they use it for cooking. ಅಂದರೆ ಕಾಯಿ ತುರಿ ಅಂತಿರಲ್ಲ ನೀವು, ಒಳಗಡೆ ವೈಟ್ ಕಲರ್ ಇರ್ತದೆ. ಕಾಯಿ ಹೊಡೆದಾಗ coconut, when you break the white color part, what you see inside, which is used for cooking, that is called cellular endosperm. Before formation of that, it will be slimy layer slimy ತರ ಇರ್ತದೆ, ನೀವು ತಿಂತಿರಲ್ಲ ಅದನ್ನು. Before that nothing will be there, only water. But when only tender coconut water is there, that is free nuclear endosperm. Then slowly some of the nuclei will be surrounded by cell wall and they become thick white color here. That is called cellular endosperm. And in coconut cellular endosperm, that white color is called kernel. Kernel, K-E-R-N-E, kernel. ನಮ್ಮ ಕಾಯಿ ತುರಿ ಒಳಗಡೆ ಗಟ್ಟಿ ಇರುತ್ತಲ್ಲ ಅದನ್ನು ಕೆರ್ನಲ್ ಅಂತ ಕರೀತೀವಿ, ವೈಟ್ ಕಲರ್. ಇಟ್ ಈಸ್ ಯೂಸ್ ಫಾರ್ ಕುಕಿಂಗ್. ನಾವು ತೆಂಗಿನಕಾಯಿ ಒಳಗಡೆ ಹೊಡೆದಾಗ ವೈಟ್ ಕಲರ್ ಇರುತ್ತಲ್ಲ ಅದು ನಾವು ಕುಕಿಂಗ್ ಯೂಸ್ ಮಾಡ್ತೀವಲ್ಲ, ಅದನ್ನು ಸ್ಕ್ರ್ಯಾಪ್ ಮಾಡಿ ಅದನ್ನು ನಾವು ಕೆರ್ನಲ್ ಅಂತ ಕರೀತೀವಿ. ದಟ್ ಇಸ್ cellular endosperm. It contains cells. But before cellular endosperm is produced, what is produced? Free nuclear endosperm. Because later, after this, it becomes cellular endosperm. Remember. Okay, so kernel of coconut is cellular endosperm. So this is about the development of endosperm. In next class we will continue this. Okay, in our next class. So keep watching my classes. Thank you.